Now that we have discussed the process of fertilization in sea urchin, we'll now discuss what is known about this process in mammals. Many of us may picture human fertilization to be something similar to what's depicted in the opening scene of the 1989 film Look Who's Talking. But the reality is that fertilization is a lot more complicated than this. To begin with, the female reproductive tract is not a passive conduit. It actually plays an active role in the meeting of the sperm and the egg. In order for the sperm to fertilize the egg, the sperm and egg have to be localized to the correct location. In mammals, the process of fertilization, the fusion of the sperm and the egg, take place in a region that's known as the oviduct. In order for the oocyte to make it to the oviduct, it has to be picked up from the ovary. That process is facilitated by projections at the upper end of the oviduct that are known as fimbriae. But in order for the oocyte to be picked up from the ovary, it must be surrounded by its cumulus cells. Once the oocyte and its cumulus cells are picked up, the beating of cilia in the oviduct and muscle contractions place it at the correct location where fertilization takes place. Now that we've seen how the egg gets to the oviduct, let's discuss how the sperm reaches the general location of the egg. And it's important to know that in humans, only about 1% of the sperm that are ejaculated, so about 200 sperm of the 2 to 300 million that are ejaculated, actually reach the vicinity of the egg. There are several processes that allow the sperm to reach the vicinity of the oocyte in the oviduct. One of these obviously is the motility of the sperm or the movement of the flagella. In some mammalian species, the motility of the flagella can actually be further enhanced through the formation of sperm aggregates. And this is common in species like the mice, where the female is promiscuous, where the female mates with multiple males in succession. And it's thought that the formation of these aggregates is a competitive strategy utilized by the sperm of different males to reach the vicinity of the oviduct more quickly. Now, I want to point out that the fastest sperm is not necessarily the one to win the race, because sperm are immature when they're ejaculated into the female reproductive tract. But reaching the vicinity of the oocyte more quickly may give the sperm a competitive advantage so that they're located in the correct region once they're capable of fertilizing the egg. Another important factor that aids in the movement of the sperm is the contractions of the uterus. These contractions allow the sperm to reach the oviduct much more quickly than they would be able to if this process was only dependent on sperm motility. Finally, another important process, and actually a very interesting process that aids the sperm to reach the vicinity of the egg, is Sperm Chemotaxis. This involves basically the sensing of a current by the sperm. The female reproductive tract has cilia that create a fluid flow, and the sperm use that fluid flow and migrate against that current in order to find their way to the egg. The sensing of that current involves the activity of calcium channels, the Catsburg calcium channels, which are the same type of calcium channels that are present in the sea urchin sperm. So this fluid current from the oviduct to the uterus gives the sperm a long-range directional cue that allows them to reach the vicinity of the egg. The sperm that are released into the female reproductive tract are actually immature. They're unable to fuse with the oocyte. This is why the fastest sperm may not necessarily win the race. Sperm have to undergo a maturation process that is known as capacitation, and this process happens within the female reproductive tract. This process allows the sperm to gain the ability to recognize the cues that are sent by the egg, to undergo the acrosome reaction, and to fuse with the cell membrane of the egg. As we'll see in a moment, some of the changes that happen during capacitation involve sperm hyperactivation, and this simply refers to changes in the beating pattern of the flagella that increase the speed and the force with which the sperm are able to move. The process of capacitation also involves changes to the sperm plasma membrane and to the acrosomal membrane. This includes both the rearrangement of lipids like cholesterol, but also changes in the proteins that are present, at the plasma membrane of the sperm. For example, one of the proteins unmasked by the process of capacitation is a protein known as sumo, which is essential for the fusion of the sperm with the egg. Although the process of capacitation is facilitated by the female reproductive tract, it is also controlled and slowed by the interactions between the sperm and the cells of the reproductive tract. Once the sperm reaches a region of the oviduct that is known as the isthmus, the sperm attaches to the epithelium of this region. And this attachment of the sperm serves to increase the lifespan of the sperm in the event that an oocyte is not available at the time of copulation. So having the sperm bind to the isthmus of the oviduct increases the chances that there will be sperm available once ovulation happens and the oocyte is present within the oviduct. In humans, studies have shown that the sperm that fertilizes the egg could have been present within the reproductive tract for up to 6 days. So once again, the fastest sperm is not usually the one that wins the race. Another potential advantage of the binding of the sperm to the epithelial cells of the isthmus is that it may help to decrease polyspermy. The binding of the sperm to this region decreases the number of sperm that reach the egg simultaneously. Studies in cows have provided support for the potential role of the sperm attaching to the isthmus in reducing polyspermy. Essentially, when this region is removed in cows, the proportion of polyspermic eggs increases. As I mentioned previously, the process of capacitation results in the hyperactivation of the sperm. Hyperactivation is thought to release the sperm from the attachment to the epithelial cells of the oviduct. And the process of hyperactivation, not surprisingly, is mediated by calcium, which leads to changes in the beating pattern of the flagella. Prior to hyperactivation, the sperm exhibit a planar movement, and once they become hyperactivated, the sperm rotate around their axis, and they also exhibit more bending. So the flagella basically acquire an aggressive beating motion. This allows the sperm to detach from the epithelial cells of the oviduct, and it also allows them to penetrate through the layer of cumulus cells that surrounds the zona pellucida of the egg. In addition to the long-range cue of rheotaxis that is provided to the sperm by the movement of the cilia, which create a fluid float in the female reproductive tract, the sperm are also provided with other cues to help them reach the egg. They become capable of detecting these cues once they are capacitated. One of these cues is thermotaxis, which we would consider a moderate range cue. Thermotaxis, as the name suggests, is the migration of the sperm following a temperature gradient. There is a 2-degree temperature difference between the isthmus of the oviduct and the ampullary region of the oviduct where fertilization takes place, with the ampullary region being warmer. Capacitated sperm are able to sense these temperature differences and migrate toward the warmer region. Another cue that is sensed by capacitated sperm and serves as a short-term cue is the presence of progesterone, which in this case acts as a chemoattractant. According to the evidence provided in a recent paper that was published after your textbook, the cumulus cells are responsible for the secretion of progesterone.